Okay, good afternoon, everybody. Thank you for joining us today. I am Jess Delfiaco. I'm the Communications Manager for the Midwest Climate Adaptation Science Center. And thank you for joining us for this month's science seminar. We're going to be highlighting the research of two graduate students who are affiliated with our network. Before we get started, a couple housekeeping notes. We are recording this webinar. It'll be posted on our YouTube page. So please turn your cameras off and mute yourselves until we get to the Q&A portion of this webinar. Let's see here. I think with that, I'm going to hand things over to my colleague Alyssa to give a little bit of background on who we are. Alyssa, go ahead.
Thanks, Jess, and welcome everyone today to our webinar. My name is Alyssa Welch, and I am the Program Manager for the Midwest CASC, based here in University of Minnesota at the Institute on the Environment. So the slide you see in front of you shows our region, and that's a big portion of how we operate and how we're organized here at the Midwest CASC. It's a regional reminder, too, of how we work with our researchers and develop research collaborations. The Midwest CASC is a partnership between federal agency USGS and a consortium of 8 institutions across the region, as you can see here, hosted by University of Minnesota. We're part of a national network of regional climate adaptation science centers, or CASCs, all of which work to produce science that informs research management and cultural resource management efforts that help fish, wildlife, water, land, and people adapt to a changing climate. Our work and the research we support centers around a set of science priorities specific to our Midwest region. And those science priorities were recently revised and launched, and you can find those on our website. We'll pop that link in the chat in a little bit. So we accomplish our mission at the Midwest CASC in several primary ways, including conducting synthesis research on relevant regional priorities, supporting a network of graduate students and postdoc fellows, 2 of whom you'll hear from today, partnering with our consortium member College of Menominee Nation to offer a tribal undergraduate research program this summer. Applications for that will launch soon. We host capacity building workshops and an annual gathering for the Midwest CASC community. We'll talk about that a little bit here later as well. And we serve as a funding mechanism with our partner USGS for climate adaptation research projects. With that, Jess, I'll pass it back to you.
Thank you, Alyssa. I'm actually going to come back to the slide in just a second. I put these out of order, but since Alyssa just mentioned it, I'm very excited to share that we have opened registration for our 2024 annual gathering. That's going to be held in East Lansing, Michigan at one of our consortium member universities, Michigan State University. And we really hope that you will all join us there, whether you're presenting research or just there as a collaborator to learn and network with folks. We want you there as part of our community. So you can hit that link to learn more and register today. It's free, it's easy. And I'll also drop that link in the chat. With that, I'm going to go back and give a warm welcome to Sarah Ramirez, who is a new postdoc who's leading one of our synthesis projects that Alyssa mentioned that we do across the region. Sarah, do you want to say just a couple words about who you are and what you're doing with the Midwest CASC?
Yeah, so my husband and I just moved up here to Wisconsin. I'm at University of Wisconsin in Madison. We just moved here in January. Before that, I had spent the past 8 years doing both my master's and PhD degrees at Auburn University in Alabama. I studied food hoarding behavior of small mammals, but I'm especially interested in how different populations within a species are adapted or conditioned to their local environment conditions, and how different populations may have different responses to things like climate change. So for the project that I am currently working on with Midwest CASC, we are interested in how individuals within populations can adjust their phenotypes or just kind of their observable characteristics to extremely variable weather conditions from year to year that we're likely to see more of with climate change. So an example of adjusting a phenotype might be initiating reproduction earlier during a warm spring to be able to match up with those earlier spring food resources. So species that can make these adjustments might be more resilient to climate change, and we might be able to identify populations that might not need as much help as others as climate change progresses. So it's still pretty early on with this project since I kind of just started, but I'm excited to see what we can learn on this topic and how we can help our fish and wildlife managers in this region sustain the populations or improve the populations that they manage as climate change progresses.
Great. Thank you so much, Sarah. Excited that we could have you on today to introduce you to everyone and looking forward to seeing how your research develops. All right, now we are going to get to the stars of today's shows. Shasta Kamara is a PhD student at the in the program of Ecology, Evolution, and Conservation Biology at the University of Illinois Urbana-Champaign. And Wendy Leuenberger is a PhD candidate at the Michigan State University. Both are affiliated with the Midwest CASC. And both are going to share a little bit about the research that they are working on with us today. Wendy, I think you are up first, so I'm going to stop sharing my slides and let you go ahead. Just so folks know, we are going to have time for Q&A after each presentation. Feel free to put your questions in the chat as our presenters are talking, or you can just come on screen and ask them when we get to that portion.
Okay, does that come through okay? Can see it.
Yep, looks good.
Okay. Yeah, everybody. So I will be talking about my research looking at butterflies within the Midwestern region. So it's pretty much the entire region that the CASC covers, which is kind of cool. And I'm looking at the long-term species as well as community trends of butterflies in this area. So the reason we're interested in butterflies and insects more broadly is that you may have seen a lot of the clickbait type headlines in the past, like 10 years or so, insect apocalypse, ecological Armageddon, things like that. But what's really going on? What we're seeing is like there's very consistent declines that are happening. It's not every single group, but we're seeing it in both protected areas and non-protected areas, and really across a lot of different insect taxa. And this is something that's pretty serious and worth considering. Insects, and I'm focusing in on butterflies, provide a lot of really important ecological services. So these include things like pollination, herbivory, food webs. So insects provide food for a lot of higher trophic level organisms. And they're also a really good indicator species. So you might get a clue of what's going on in the ecosystem on insects before you would get such indicators on something like birds, just because of that different trophic level. And people also care a lot about butterflies as well as pollinators in general. And it's really neat to see that people engage with them, planting pollinator gardens, doing the citizen science that this research is based on, things like that. So they're a really good group to look at to see what's going on and to understand how ecosystems are affected. But it's a really hard type of question to ask and to answer. So we see a lot of extremely variable patterns across space and time. So you might have like boom years and bust years for a lot of insects. You might have something happening in Minnesota, but something very different in Ohio, things like that. And it's really hard to get data sets that are long enough to kind of tell what's going on without like taking those fluctuations, being able to account for those fluctuations as well as what's going on in a larger region. It's also really difficult to look at both species specific dynamics as well as overall community dynamics. But we're able to get at a lot of these key issues in the Midwestern United States due to these 5 citizen science programs. So we have the North American Butterfly Association, and then we also have 4 state-specific monitoring programs in Illinois, Iowa, Ohio, and Michigan. So this is really cool data. It's some of the best in the world. So I'm really excited to be able to share with you what we're able to find with these data. These data are already being used. So the Ohio data in particular was published in this really cool study that looked at declines over the past 20 years, but really restricted to that Ohio data set. It's super useful, but I think we can build on it. So what this study was able to show is like we are seeing declines in butterflies in the Midwest. So I expect that I'll find that in my research as well. The plot on the left is showing like the overall trend over time, just compressing all of those species into one. And then you can see the species specific patterns here on the right. You can see a lot of them are declining, but insects are really variable. So you see that, it's some species are declining, some are staying the same, a few are increasing. They were able to estimate trends for 81 out of about 120 species that they had that are in Ohio. So there's a lot of species that aren't able to get trends using these methods. So there's a lot that we think we can build on with my research. So kind of recapping those goals and the motivation, we've got these butterflies as a really key group to look at insect declines and to understand some of these ecosystem services and what's going on in the system. We have data from these groups that are available 10, 20, kind of even 30 years ago. I'm kind of limiting it to 20 at the time. And then we've got current analyses that are looking at these. It's really valuable, but they're really restricted to these single species models or limited spatial extents. So the goal of my research is to be able to get abundance trends of more species and overall communities over a longer time period. And the goal is to get as many species and as many years as possible. So what these data look like throughout the Midwest, we've got kind of this spread throughout. So the darker purple dots are those North American Butterfly Association, and then the brighter ones are the state-specific surveys. They'll go outside of their state a little bit, but they're definitely more regionally restricted. And with it being, volunteers collecting the data, you can kind of see where people are located. So Chicago, Cleveland, things like that. You end up with more data where people are. And that's where those North American Butterfly Association surveys are really helpful because they're pretty well spread out over space. So what happens to get the data is different state transects as well, and then the NABA are kind of a different type. So volunteers go out to certain sites and walk the same transect anywhere from once a week throughout the course of the year or like 6 times over the course of the year. Usually from about May to September, but some of them are more restricted. So this is really valuable data. It gives us very structured data, so we know both what species are there as well as what species weren't seen. So that's really useful to have. Those North American Butterfly Association surveys are more of a BioBlitz type event. So volunteers go to like a 25-kilometer circle and they go to different parks within it. So have some people at one park, some people at another at the same time, just counting as many butterflies as they see in 24 hours. So that's also really useful data. And these go back for quite a few years, depending on the data source. The NABA started as early as 1975, and Michigan's a bit newer at 2011. There's about 130 species in this model or in this data set, but that's too many for like one model, even though we're trying to get at some of these like community dynamics. So I was able to split up these species into smaller groups of like 20 to 50 species. And we wanted to do this in a way that like would be interpretable. Luckily, there was this really recent data set that came out that catalogs all of the traits of butterflies throughout the world for any species that there's data of. So I was able to use this to create 4 different groups for my modeling. So I first split them up by whether they were a migratory species like the monarch butterfly or a resident species that stays year round in the same area, or maybe they might move around a little bit, but they don't leave the area. Then for the residents where there's more of, I split them into the ones that have 1 generation per year or multiple generations per year, which is pretty common, where you have like maybe a spring one coming out and then later in the summer you have another generation. And then because there were so many of those multiple generation per year residents, I split them up. So in green is the ones that the caterpillars can only eat 1 host plant family, and then the generalists can eat multiple host plant families. So that's just a way to kind of break these into groups that were meaningful, but also giving us kind of these species specific information. So I'm going to go into the type of modeling a bit. I'm in a quantitative ecology lab, so this has been a really cool thing to be developing and something that is kind of a broadly applicable framework. So it's called an integrated community model, and the integrated means we've got these multiple data sets. We have the 5 different volunteer programs, and the community refers to us having multiple species in the model. The fancy word, being a negative binomial hierarchical Bayesian model. So a lot going on here. And I'm doing this separately for each of those functional groups. And basically the framework from this model is trying to explain trying to figure out how many butterflies are actually there. And this is an index, so we don't know the exact number, but we can get something that gives us insight into those trends and those amounts. And we're trying to do that based on the number of butterflies that the volunteers went out and actually saw and put on their data sheets. So we've got a few different steps in the middle. We have to figure out like the mean count versus the observed count. And then there's a lot of variability. You'll have a lot of zeros, a lot of ones, and then sometimes you see 1,000 cabbage white butterflies. So just different things in the model to account for that. We have different covariates, and they broadly fall under these categories of space, survey, species, and time. Space and survey are things that we know affect butterflies. We know these things are important for understanding the data and explaining the data, but I won't actually be going into like the effect that any of these have on the trends over time. So we're looking at controlling for things like which county we're in, what site we're at, what amount of open cover or crop cover, which of those programs, just they have different protocols. So we have to account for that if there's more effort, more surveys, things like that. But I won't be coming back to these. More of the meat of the model comes to the species and the time. So for species, we have this in as a random effect. This is what lets us estimate for multiple species at a time. This also has a really beneficial like aspect to it in that because we're drawing these species like from a distribution, we're able to estimate rare species that we wouldn't be able to do in a single species model because we can kind of borrow power from those more common species. So that's a pretty useful part here. And then for time is really the piece that's going to get us at things. And we've got a few steps here. So the goal is to get a week and week squared term, and that gives us like an activity curve. So butterflies aren't going to be active from May to September, but they're going to kind of peak somewhere in the middle. And that's what lets us kind of model that. And then we also have year and species in year terms. And so basically what that does is lets each species in each year have a different activity curve. We then use this in data in another step post hoc to determine those relative abundance indices based on the area under the curve. So for example, just to kind of walk through all of that, because I know it was a kind of hard to visualize statistical stuff. Here we've got our week over the course of the summer and kind of a relative abundance index at any given point. For each species, we get this sort of curve over time of here's when they're most active, here's when they're least active. We can take the area under the curve, and that gives us a relative abundance index for that year for that species. We then have these for all of the species in our model and all of the years. And these areas under the curve are what we're actually able to estimate the trend on with just a regression framework. So kind of to recap it, this model, I have a total of 122 species, which is really cool. So the previous study that we're kind of like comparing to that was really great was able to get 81, and we're able to get a lot more here, which is cool. I've got a time period from May 1st to September 30th, which really covers most of the butterflies that would be active in the region. This is 22 years. I've been pushing forward with this model to get back to 32, but I don't have those results quite ready yet, so they're not in here. And then one of my lab mates created a package, the SP abundance and SP occupancy, that have been super useful for this project. So shout out to Jeff Dozer if you ever have stats questions or models like this. So I'm really excited to have some results. Like I said, they are a little bit preliminary, but I think the general trends are going to stay the same. So here we've got the species on the x-axis, and I don't have the species names here because there's 122, so it's not a good way to piece things out, but I'll go into detail on a few more of them. And then we have the annual trend as the y-axis. So anything that's below the line means that they're decreasing over time. Anything right around that zero line is stable, and anything in the space above is increasing. And then I have those different functional groups that I mentioned earlier, just being the different boxes. So what we see is pretty much what we expect. We do have more species declining than increasing by quite a lot, but there is a lot of variability in what's going on with these trends, and there are quite a few that are close to or overlapping zero. So they're not strongly negative, but there are a lot that are declining pretty dramatically. I'm going to go through a few species that I think are really helpful for kind of diving into what's going on here. So the first one I'm going to talk about is the regal fritillary. This is a species that's of conservation concern in the region. So there's been management happening in order to conserve them. And what we see is that they do actually have a really flat trend. So they're not increasing, they're not decreasing. And that's pretty good news for a species of conservation concern. being managed given the context that most butterflies are decreasing. So here we've got a flat trend, pretty much an even 0.46 chance of whether they're increasing or decreasing, and a very slight increase over time. There's some species in there that were increasing. So here's the sleepy orange butterfly. So it's increasing by about 2% per year. And that makes a lot of sense in context of this species because what we're seeing is that they're moving north with climate change. So we would expect to see them increasing as they kind of get into new areas. And it doesn't necessarily tell us what's going on with the entire range of this species. So we are kind of limited to the Midwest in that way, but it's good to see the variation in what's going on with different species. Other species are declining, one of those being monarch butterflies. Monarchs are probably the most well-studied butterfly by, I think, a pretty wide margin. So there's lots of different trend estimates, but this is pretty in line with what we see in different studies. So that's good to see in the sense of the model seems to be doing a good job or being reasonable. We're still kind of working on the trend to finalize it. So this isn't something set in stone by any means. But also, yeah, they're decreasing. That's unfortunate to see. It was interesting to me that one of the species that was decreasing the most in contrast was the cabbage white. And so this is a species that's not native to the area. So in some regards, and it's a pest species on agricultural crops. So in some regards, it's like not a bad thing that it's decreasing, but also if you think about the amount of butterflies out there and the biomass that like a really common species provides in terms of those food web services, pollination, things like that, it potentially has a really big impact. And what we're seeing is we seem to be seeing more common species declining more dramatically than the rare species, which is not unique to this study by any means. So kind of some preliminary thoughts because we're seeing those like declines in the common species. I don't have richness and diversity to present to you now, but the preliminary stuff shows that like diversity is increasing because the common species are becoming a little bit rarer, more even proportions across the different species. And richness is decreasing a little bit. So by about an average of 2 species over about like 30 years or so. So those are just kind of sneak peeks at what the model is going to be able to give us, but I don't quite have finalized enough to be putting on a graph currently. So kind of going into what does this all mean? How does this fit into other projects? It makes sense that we were able to see butterfly declines across the Midwest, given that that's what we're seeing in parts of the Midwest and parts of the world, various situations. There's a lot of reasons insects are declining. The Midwestern United States, has a lot of agriculture, and that can be very challenging for insects when you're dealing with things like insecticides, pesticides, things like that. This model was able to get us a lot of new information about species trends. So we were able to get almost all of the species that we had data for, and that's novel information about several of them. There's declines across the different functional groups and in common species as well, but not all species are declining. So yeah, I kind of touched on this already, but we've got a lot of different reasons that insects are declining. And there are some conservation efforts happening. We work with some people that are affiliated with the Midwest Cask or else otherwise other parts of the nation in terms of, okay, here's the trends and can we have people that we can work with to translate that into what we do next? And the Midwestern United States, the agriculture that I mentioned makes it a pretty tough place to be. Lots of other things going as well, like land cover change, things like that, climate change. This project's still under development, so I'm incorporating some more recent data ending at 2023, as well as pushing it back a while and getting at those community trends that I mentioned. Hopefully in the future, I'll also be able to look at the relationships between butterflies and climate. So trying to get at what's going on there, why are these changes happening, and then what does that mean for the future? What do we think will happen moving forward? That stuff's a little ways down the line. I don't have that in the workflow yet, but hopefully in the next couple of years or so, that'll be developed. So it's been really, really cool project to work on with lots of different people. I want to send out some acknowledgments. So lots of really awesome co-authors that we've worked with here from MSU people as well as agency and other universities as well. This project would absolutely not be possible without all of the volunteer hours that have gone into collecting these data. I did a summary on the data, and I have about 90,000 hours of volunteer time in here. And that volunteer time does not include anybody learning how to identify butterflies, entering data into a spreadsheet, traveling to go do these transects. And it's really incredible what that all turns into in terms of us actually being able to estimate these really good trends for an entire region of the United States. And for context, that 90,000 hours is basically 24/7 for 10 years, or else somebody's entire career from like graduating undergrad through retirement. So it's really cool to be able to work with these data, and I hope to talk to a lot of these groups to show them what we were able to do with it and see what they think about all of it. Also, a lot of different funding sources, including the Midwest CAS, contributed to that. And then the picture here is a citizen scientist in Nebraska who's not in my data set, but he is my dad, so it's a fun one to include up here. And that's what I've got. So if there's any questions, I'm happy to answer them.
Thanks so much, Wendy. That's a wild stat on volunteer hours. Really drives home the scale of this work. We do have a couple questions in the chat. We've got one from Sharon Stevens, which is about asking if you have looked into the endangered Karner blue butterfly at all. Their habitat is primarily oak savanna and lupin. And then there's some further context in there about work being done to conserve the Karner blue. Any thoughts there?
I haven't looked into that species in particular yet. So hopefully when I get this published, hopefully submitting by the end of summer, we will have trends for every species in there. But feel free to email me and I can let you know and look it up, see what my results are indicating for them. I just don't know it off the top of my head for the species. I think they're not in most of these areas because they're in New York a lot and then they're in like Minnesota, but I think they do show up in the data set. I'm just writing that down also.
So we've got another question. Working within the oak openings region of Michigan and Ohio, are there any species or initiatives that folks should be tuning into as far as conservation efforts?
That's a good question. I know that in some areas further south, there's been a lot of don't mow early spring type things, and there's various like habitat things. I'm not super keyed into the conservation actions right now. I've been very focused on the modeling effort. Um, so yeah, my apologies, but I don't have a great answer for that one.
All right. Anybody else have a question for Wendy? Feel free to go off mute and ask your question directly, or again, pop it in the chat. I'll give it another minute.
Hey, that was a great presentation. I was curious about you had your four groups that you delineated. It seemed like they had about the same trends, or do you think that like some are a little bit more resilient than others?
Yeah, that's something that we're looking into more. I had them organized by the group that was declining the most was the specialists, and I kind of had expected it to be the migratory group instead. It was interesting to see that like overall, the average for migrants was actually a little higher than the other groups. So yeah, it's interesting. I don't quite know how predictive that is. So a lot of the other research shows that like the traits aren't usually super predictive, but they can give insights. Some of the other things I'll be looking at is like the species that overwinter as eggs versus larvae, pupae, or adults. So the ones that overwinter as eggs are sometimes a little bit more vulnerable. So it'll be interesting to see if like that pattern is seen in my data. But yeah, it's pretty across the board and all over the place in terms of who's declining and increasing.
Okay, I think we will switch over to Shasta now. I'm sure if a question pops up, Wendy will be keeping an eye on the chat and could respond to you there, and we might have a few extra minutes at the end for further Q&A.
Thank you all.
Alrighty. Perfect. So if everyone can hear me and everything's good, I'll go ahead and talk a little bit about some of the work that I've been doing recently with this really cool fish, some of the management challenges that it currently faces, and how my projects are hoping to fill in and address some of those gaps. So starting with fisheries management, it's kind of the paradigm in which my work falls in, where fish populations are kind of the focus here, and you have a bunch of different parts influencing these populations. So starting with fisheries users, traditionally who you were thinking of like people fishing for fun or going out and fishing for commercial harvest for a living or for cultural practices. And each of these different users might have different reasons for either harvesting or releasing these different fishes and why they are going out and catching them in the first place. And then this all in turns influences how these populations are doing. So what the makeup of these populations are and their status, and that can influence the catch rates that these fisheries users are generally seeing. And then you have the other side with fisheries managers, who are the ones that are responsible for ensuring that these fish populations can support all of those different uses from those different fisheries users, and that they're healthy not only now, but in the future for future generations to use these resources. And so fisheries managers often implement things like regulations or stocking to, again, ensure those populations are healthy, and they're able to monitor these populations with a lot of different traditional fisheries techniques. And of course, this is a socio-ecological system where people influence the fish and fish influence the people. And the people also influence the people here. So there's a lot of moving parts with fisheries management and a lot of different strategies for ensuring that these populations are doing well, including the incorporation of things like scientific data. So making sure that we have the most informed management to ensure that these populations are doing well. Fisheries managers might collect their own data through either fisheries users or traditional fisheries techniques, but also through collaborations with outside agencies like academic agencies or academic institutions, like how my project has kind of come to fruition. So one cool case study we can use here is the American paddlefish. This is a really large riverine fish. You can see in this picture some anglers that had one of the more recent world records. It's primarily a riverine fish that is really popular among anglers because they're fun to catch, fun to fight. Some anglers will harvest them for meat, and they're also really important in the commercial fishery because of roe or caviar. And so this fish has a mixed use of different fisheries users, both commercial and recreational. And these fish often span pretty far, so anywhere from southern Canada down to southern Louisiana, all throughout the Mississippi River drainage basin. And they are listed on the IUCN list currently as vulnerable, but this varies by state. In some states, they are extirpated or they have very low populations and are protected. And in other states, they're managed as a sport fishery or as a commercial fishery. So we have a lot of different regulations that these fish can experience. And I wanted to take you through, so if you're a paddlefish and you're caught in either a recreational or a commercial fishery, there's a different paths that you could take and different fates that you could experience. So just looking through commercial harvest, if you're caught in a net, which is typically what these commercial fishers are using, you might end up being a male paddlefish in a roe or caviar fishery, and you might end up being released because those commercial harvestmen don't necessarily you don't have that product that they want. And so once you're released, you can experience two different fates. One, you could survive, go on, do great paddlefish things, reproduce and go back to that population, or you might unfortunately experience a cryptic mortality event. And it's cryptic in the sense of we release that fish back into the water and hope that it does well and reproduces, but in some cases it might experience a mortality anyways, but we are unable to often account for that. Another fate that you might experience is being under that regulation size that those fisheries managers have set. So if a commercial harvester catches you, you have to be released in order to be compliant with that regulation. And again, you might survive, which is the goal, or experience a cryptic mortality event. And finally, you might be of regulation size. That harvester wants to keep you, and so you're harvested. Or if for some reason they don't, again, you could be released, survive, or released and experience that cryptic mortality event. So overall, we have a lot of concerns with our fisheries and ensuring that as many fish as we put back are able to survive and continue on. But we want to know what kind of influence other things like environmental variables might have on these fisheries. And this is where looking at our harvest seasons kind of comes in. So in Illinois, we actually have commercial fishery and a sport fishery. And here I'm highlighting in this graph our water temperatures for the upper Mississippi River during our commercial seasons. And so these two dashed lines signify the bounds of one season, starting in October 1 and ending in May 31. And so if you're a paddlefish and getting caught in these nets, you could be caught and released and expected to survive anywhere from 22 degrees Celsius all the way down to 0. And so we have a wide thermal variation that these fish are experiencing on top of that stressful event of being captured in the first place. And we currently have pretty limited information on paddlefish like thermal tolerance and how they respond to these stressors at different temperatures. So our management decisions can't be the best that they could be if we had that information. And so my projects are aiming to quantify paddlefish thermal tolerance and response after a simulated capture and release across a range of temperatures using a couple of different approaches. So first in the lab, we were assessing paddlefish performance after exercise at different temperatures and thermal tolerance. So how well do you perform at these different temperatures, but also how much of that upper heat tolerance do you have when you're acclimated to different temperatures? And then in the field, we wanted to evaluate post-release behavior after a simulated commercial capture event. So after you're caught in a net and released at different temperatures, how do you behave and where do you kind of go from there? So starting in the lab, looking at our simulated capture and release methods, we acclimated juvenile paddlefish to 3 different temperatures, 13, 17.5, and 22 degrees. And then after that 2-week acclimation, we exercised fish individually in a pool, as you can see in this diagram, by chasing them and pinching their caudal peduncle to kind of simulate that exercise event, either if you're being angled and fighting an angler on the hook, or if you are getting caught in a net and trying to wiggle your way out, expending that energy. And then afterwards, we took those fish and allowed them to recover individually for either 30 minutes, 4 hours, or 8 hours. And that's to get an idea of their recovery over a span of time. We also had a group that we chased and then immediately sampled, and a control group that we used that was never chased to serve as our kind of baseline levels of stress. And then we sampled them. We collected blood from their caudal vein and spun that down to get their plasma. And we wanted to look at secondary stress indicators. We used plasma lactate, which is actually common in the fisheries world as a secondary stress indicator. And if you think about it, if you go and run around, you build up lactic acid in your muscles. It's the same thing, but for fish. And so looking at some of those results, we have plasma lactate along the Y and the recovery group along the X here. Our coldest fish are in these clear bars. The gray bars are our middle temperature fish, and then our warmest fish were in these dark colored bars. So looking at the clear bars first, we can see that we have fairly low levels until about 30 minutes post-release or post-recovery. And that kind of indicates that we have a peak in plasma lactate here, but we're back to baseline levels within 4 hours and continues to drop over the course of 8 hours, or the duration of our study. For our warm fish, our middle temperature, we can see that there's a very similar pattern. However, this peak is not significant, but then we go back and decrease over time. And then our warmest fish, we can see again a peak at 30 minutes. However, that peak never drops throughout the rest of our study, or those 8 hours. And so our warmest fish weren't able to recover even after 8 hours, even though our coldest fish did. And so this is kind of indicative that these warmer temperatures aren't necessarily the best for our fish to be able to recover at. And so this might have some implications for our harvest seasons. And then we wanted to look at thermal tolerance, or our upper tolerance to temperature in these fish, because this is a common metric that we know for a lot of other species, but we don't know for paddlefish yet. And so again, we were acclimating to 13, 17.5, and 22 degrees. And we placed 10 of those fish per acclimation temperature in a pool, and heated them up from acclimation temperature until all fish lost equilibrium and were removed one by one after that. And this is a great metric because it's not lethal, but we're able to kind of say, okay, that is the most upper temperature that they could tolerate before they can't do normal fish things anymore. Because when you lose equilibrium in the wild, you're more vulnerable to things like predation, and the likelihood that you'd be able to recover is pretty low if you weren't in a lab setting. And so looking at some of those results, we have loss of equilibrium temperature along the Y, which is akin to upper thermal tolerance, and then acclimation temperature group along the X. Focusing on our purple boxes first, we can see that our coldest fish tapped out at around 28 degrees, which is much cooler than our warmer two groups. However, our two warmest temperatures did not differ in upper thermal tolerance from each other. So this kind of indicates that these fish might have a concrete ceiling to upper thermal tolerance, which could be concerning because even though you might be in warmer and warmer waters, you might not be able to take those heat waves or temperature spikes that might occur. And looking at our gray boxes, this kind of further supports our thoughts here, as these are acclimation temperatures. And we were able to see the change in, from acclimation temperature up to our upper thermal tolerance. And we had decreasing ability to go up in temperature as we move to warmer and warmer groups. So again, your capacity to tolerate heat kind of diminishes as you are acclimated to warmer temperatures. And so what have we seen from our lab studies thus far? When evaluating the performance of paddlefish, we saw that paddlefish above 20 degrees don't seem to recover from a simulated exercise event, even after 8 hours, unlike our cool fish, which were doing a little bit better. And then trying to determine that upper thermal tolerance, we saw that paddlefish might have an upper thermal limit, with thermal tolerance not increasing with fish held above 17 degrees. So this, again, is kind of concerning for our warming waters. Moving on to the field. So we had our post-release methods here. We partnered with the Illinois Natural History Survey and Oklahoma Department of Wildlife Conservation, who were doing catching fish either through their annual surveys or through partnering with commercial carp fishermen who incidentally catch paddlefish. And so in both cases, they're using these large mesh gill nets pictured here. And once fish were collected, they were brought into the boat, measured, weighed, all the normal things. And then we attached a small device known as an accelerometer with a Velcro belt to the caudal peduncle of these fish. And it was further attached by a length of braided fishing line and to a float. And these little devices are able to track acceleration in 3 different axes. And we're able to sum that up into a metric, which is basically just how much is that fish moving around, otherwise known as overall dynamic body acceleration, or ODBA. So we would release this fish with that device for about an hour and then would retrieve it after that monitoring period to get an idea of what these fish are doing after that simulated capture event. So looking at some very preliminary results, we have mean ABA along the Y, and then time bends, 10-minute bends along the X. So our pink color for this bottom line is our Upper Mississippi River samples. The purple is a lake known as Grand Lake in Oklahoma, and then another lake, Keystone Lake in Oklahoma as well. And so the Mississippi River fish were sampled at around 3.5 degrees Celsius. Grand Lake was sampled at around 10 degrees, and then Keystone Lake around 14 degrees. So the first notable thing here is within their specific sites, which happen to correlate to temperature in this instance, as we get more data, that'll kind of go away, but they are different at each different site. So generally that trend is pretty stable within each one of these sites. But we also see that we are seeing differences between time bends. So in both the case of Grand Lake and the Upper Mississippi River, we had some variation in the beginning of how much they're moving around, but both seem to have this instance of reduced movement between 31 and 40 minutes. And a little bit of speculation here, if you think back to that plasma lactate graph, that spike was at around 30 minutes. So potentially they're reducing their movements to kind of conserve energy and put towards recovery, but we don't see that in the Keystone Lake fish. So maybe this is a delay with temperature. We're not really sure. But more data as we continue to collect it will probably help to determine what kind of trends we're seeing once we release these fish. And so what are the kind of management implications here? Again, we wanted to quantify the response of paddlefish to simulated capture and release across a range of temperatures in relation to those harvest seasons. And we saw that 20 degrees might be a threshold for paddlefish performance. So these fish above 20 degrees don't seem to recover from a simulated exercise event even after 8 hours. And so this can make them more vulnerable during that time period. And we see that thermal tolerance might have an upper limit, with fish above 17 degrees not gaining any more thermal capacity to tolerate those upper temperatures. And for our overall dynamic body acceleration, we saw that it differs between time bends, but as to what trends we're seeing, we're hoping that these warmer temperatures, once we get those data, we'll be able to provide some more insight as to what's going on here. And this graph is just a reminder of how much of these seasons do overlap with that vulnerable temperature. Currently, it's not a lot, but as we know, climate change has been shifting our water temperatures, so we might be seeing earlier warming and staying warmer later. So we've got to be mindful, at least on the commercial side, that the edges of our seasons are going to be more vulnerable. But we also have year-round recreational angling in Illinois. So these peak temperatures in the summer, you still might be able to be caught by recreational anglers. So it's still important to take this information in mind when making some decisions for these fish. And with that, thanks for listening. I want to thank Jackson Galam, pictured here, for helping collect a lot of this data. And this is funded primarily through the Illinois Department of Natural Resources and U.S. Fish and Wildlife grant through the Sport Fish Restoration Fund. INHS and Oklahoma Department of Wildlife Conservation allowed us to come out and collaborate to collect a lot of that post-release behavior data. And then, of course, for the Midwest CASC for inviting me to speak today and funding me and my research doing all this. So with that, I'd be happy to take any questions.
That's a really cool project and neat to see the accelerometers. I usually think of them on like elk and not on fish. I was wondering what the fate of the females is that are the roe that go through the roe fisheries. Like, are they able to be released and survive?
So generally, I haven't gotten to speak directly with any commercial paddlefish harvesters, but it's generally a lethal process. They'll take them and process them, and in some cases they might use the meat for other things, but no, those big females are generally taken out of the populations.
So how far away do they go in 1 hour with that thing attached to them?
Yeah, it's really funny. On the bigger lakes that we were doing in Oklahoma, we would lose a fish for a good 20 minutes because they will shoot up or downstream relative to where we are. But some others will just kind of meander across the channel and just sit there. So it's kind of varied a good bit. And we're hoping to see that a little bit in the data of maybe by temperature, maybe they move around a little bit more. Because all of these temperatures we have right now are fairly cold. We had up to 14 degrees and our lowest in the lab was around 13. So they're still pretty cold. I think once it warms up, they might be moving around a little bit more.
Okay, I'm having a really tough time typing all this out. I This was amazing, and I have so many different questions that are really just not even within the scope, I think, of what you were doing and where it might be applicable. So I'm wondering, you know, uh, thinking big, um, who all might care about these findings and future findings, and how might they be used?
Yeah, so I think that honestly, the managers are going to be the biggest ones that are interested in this. So this was really a manager-driven project from the Illinois Department of Natural Resources, which is really cool to know that they're interested in trying to figure out, should we be setting our seasons by date, by water temperature? When are they most vulnerable? How can we shut down a season or warn anglers, oh, it's a hot day, maybe try not to fish today. And we have a lot of interest in snag fisheries. So these fish, you can't catch them by just putting a hook in the water and hoping they bite. They're filter feeders, they don't do that. So we have a big snag fishery in Oklahoma for sure, but a lot of the northern states, Missouri, North Dakota, South Dakota, Montana, all I know have big snag fisheries that also have snagging guides. And so guides really want to be able to keep their business open, but they don't want to lose fish if they don't have to, right? So they might be a little bit more mindful of taking people out on certain days if we're able to say, okay, above this temperature in this region, you should be conscientious because it's going to stress the fish out a little bit more. And anecdotally, even people that sample paddlefish know like above a certain temperature, they just don't recover as well. They don't go and swim around as much. So I think that the managers and the recreational users are going to be our biggest proponents. But of course, that all relates to our commercial harvesters too, because if they are not recovering or experiencing mortality events from other fisheries, that influences their ability to catch them commercially as well. And they want to be able to persist that fishery.
Okay, well, I'm going to pop up a quick closing poll, but while that's up, y'all are welcome to ask any final questions of Shasta or Wendy. Um, thank you guys so much for great presentations. It's so cool to hear about your work. And thank you to everybody for joining us today. Let's see, here's